I feel like a little bit of an intruder, but I always feel like a little bit of an intruder, but probably more so now than mostly. But actually, I think it's really nice that I've got the opportunity to talk with you. I've enjoyed listening to the talks the past few days. And of course, the field of DNA nanotechnology is coming on in incredible ways. What I'm going to try and talk to you today over the next 25 minutes or so is about thinking about the origin of information in biology and in chemistry in general. So to some degree, it's a complete segue. I hope I don't bore you all or confuse, or even worse, confuse you. But what I hope I do is explain to you some themes that then will come together at the end, a new type of kind of chemical informatics, what might be interesting for those of you interested in bioinformatics. And I'm assuming you can still hear me?
Yes, great. Yes.
And you can see the slide. Good. Okay. So look, so I kind of want to be provocative and suggest a few things that to understand chemistry and also the origin of life and also how information gets into biology. In fact, what information is, we need to go all the way back to the beginning. And a couple of things I want to say is that I think the reactions don't exist, right, outside of human kind of culture and contingency, but reactivity exists and chemists exist. The other thing is that chemistry is not complex as a given, it's combinatorial. And it seems that biochemistry inserts bias into our chemical space. So what I'm trying to say is that chemical reactions, as we accept them, those of you that are chemists and some of you that rely on chemical reactions, are actually something that's invented on Earth by evolution and lately by chemists. But the thing is, chemistry is not magic. There is no predisposition. A lot of problem with the origin of life is people require RNA to magically be invented. And that's why I gave Thomas not a hard time, a trigger warning on emergence, because people basically, it's almost like there's random stuff in the universe and things emerge. Well, no, there's no randomness in the universe. Randomness does not exist. There are pseudo-random things in the universe because it's really complicated. And we might argue about whether quantum mechanics is a deterministic theory and whether there's randomness there, but it really doesn't matter for today. I think I can convince you that chemistry isn't magic. And if I can't convince you right now, I will in 2 slides' time. So I want to first just say, look, chemical reactions are invented. The key points for the talk are that reactions are not random, they require information. And I'll explain why that is. And I'm also going to present a theory called assembly theory that helps explore the emergence of evolution. So there the emergence word is. And then having set up those 2 points, I think the point that may be useful for this community is to start thinking about genetic information using assembly theory and assembly spaces. And I'll get to why that might be interesting, but some of you might be able to tell me in fairly short shrift, give me fairly short shrift and tell me why it's wrong, or maybe why it might be useful. So let's go on. I'm going to talk about in the first part for and I'm okay to speak for about 20 minutes from now. Is that right? To get us finishing roughly on time?
That's correct. Yep.
Well, I know we start a little bit late, but I think we can do it in 20 minutes without being too rushed. So I'm going to talk to you about chemputing, which is this idea of running code on a robot to make a molecule. It's not a molecular computer. I'm not really qualified to do such things. There are many better qualified people who've been doing exciting stuff like here today. But what I want to kind of ask is, what is information and why do you need it to navigate chemical space? And information can be quantified if you want to use Shannon entropy. I don't particularly like the use of Shannon entropy, but it is a way that you can use it, and you could use it in chemistry. You could look at also Shannon entropy to kind of articulate how difficult it would be to navigate a tree of dice throwing. Or take the vitamin B12. Vitamin B12, some chemists claim, just forms randomly, but yet the total synthesis of vitamin B12 required 91 postdocs, 12 PhD students, 72 steps. And so I find it interesting that we are able, that vitamin B12 can be discovered by the universe, but somehow takes a long time for chemists to master it. And likewise, in that movie, I don't know how well the movie is flowing over Teams, but that was the assembly of the molecule Taxol. And so at this point, I'd like to pause and say, okay, if we go to Mars, there's a new rover on Mars, and that rover could find a milligram of B12 or a milligram of Taxol, what would you conclude? Would you conclude that it just randomly formed there? Or would you conclude that an informational process had to somehow construct it? And I think hopefully I might kind of convince you it's the latter in the next few slides. But wait, chemistry exists. So where's all the information coming from? Let's just take a current, people thinking about how we might get to RNA base pairs and using hydrogen cyanide. If you look here, we've got this adenine that's been synthesized in substantial amounts by heating a solution of hydrogen cyanide in aqueous ammonia for 1 to several days between 2700 degrees. And then there's a load of extraction that goes on there. So even that make like easy or prebiotically plausible, the chemist has to go, and if you view the chemist like a bit of an analog machine, they have to have some information and some way of adding those reagents together in a flask to exploit the intrinsic reactivity of hydrogen cyanide and ammonia to produce these molecules. So it does seem that laws of physics and chemistry do encode some information, but there's something missing, isn't there? That we can have simple reactions producing simple molecules, as you can find in space. C60 was not made by a robot or an alien. It was just made by, pulverizing carbon with energy. And there is a nice trap that makes C60. Likewise, you can make formaldehyde or other simple molecules, but they are simple and therefore statistically easy to imagine combinatorially that they would get produced. Kind of with this in mind, in my group a few years ago, we'll be inventing a kind of what I would call a chemical state machine that basically allows us to take frustrated with organic chemists not telling me the truth about their reactions, and every time I try to reproduce something in the literature and it not working, I decided I'd make a robot to do it. And what we were able to do is to show that we can abstract chemical assembly into a state machine, and that could make any molecule material. All the chemists at the time told me I was talking nonsense. It wasn't possible because chemistry was somehow magic. Well, chemistry isn't magic. It's hard. And organic chemists are extremely well trained and extremely talented. But if you make the right abstraction, you could turn organic chemistry into abstraction of 4 things: the reaction, the workup, the isolation, and the purification. And you could build a machine where you could change its states and input reagents and pass reagents through that machine and make molecules. And we made, in this machine, we've made Viagra and Nitol and refinamide. And using this very same machine, we've now done 100 different molecules. And there's nothing magic there. I mean, the machine often breaks. But what I want to get through here is that one machine, many different configurations, many different reagents, you can make the molecules. And the complexity of the molecules, if you like, can be related to the way the complexity of the program and the requirements. To do this, we had to make a language. This is called KDL, chemical computing language. You can see on the left-hand side there that it looks like a normal kind of almost like a script where you define the hardware, the reagents, and the procedure, and then you have a graph of that, and then the graph gets run on a robot. So you could almost view the robot like a CPU, and the graph like the microcode. And the input on the left-hand side like a program you put in, and that program is then run and compiled. And we say it's chempiled because the program is not run on silicon; it's actually run physically. So there's a kind of a weird kind of machine in that it's not a digital computer, but it is a chemical state machine. And we've been able to use it to make lots of different molecules. We can make peptides and we can make DNA. And the interesting thing now is we can program, and this might be of interest to the DNA nanotechnologists, is that in this machine, not only can we make DNA, but we can add on any chemical that you'd like into that DNA and make modifications of the bases and wire those in. So we could go beyond normal DNA nanotechnology with this programmatically. And that's, I think, a really exciting event that will happen in the next few years, hopefully. And what I've shown on the left-hand side up here, if you look at A, I've got all these unit operations. How we managed to do the coding is you've got the unit operation add, separate, filter, dry, evaporate, deoxygenate, and these are programmable functions, okay, where they're repeated again and again. And so if you do that and wire them together in the right way, you can do reliable syntheses. Up the top here, we're doing a cross-coupling reaction. In the middle, in B, we're making a diazurine. And the bottom here, we're making a peptide. And so in this way, we're able to kind of, in the meaningful sense, I think, of the word, and the way this community uses the word programming, you definitely do program DNA and shapes of DNA and tilings. A lot of supramolecular chemists and systems chemists talk about programming chemistry. What they really mean is going to a lab and dumping some stuff in a test tube. I'm not really sure that counts as programming because I don't really view the test tube yet without a universal standard as something that I could defend that programming, but you know what I mean. So we're getting there. So I've done part one, which is to say, okay, chemistry reactions require information. Otherwise, you just make random stuff. Unless it's very simple. What do I mean by simple? Can I quantify in a reasonable way what I mean is a complex molecule? And the problem with complexity science, the science itself is quite complex because people discuss complexity, whether it be computational complexity or some other measure of complexity, whether we say start to measure entropy. And the problem is that we get stuck because we talk about disorder or complexity. But really, when we're talking about making stuff and building stuff, we're talking about assembly. And so what I've been doing for a few years is try and ask if we can instead express the information required to build something if we understand the building blocks and the processes to make it using a new type of approach that we call assembly theory. Assembly theory is different to complexity theory, because we're not talking about, because complexity theory is not really materially objective. And I'm happy to discuss that, argue with you about that, because it's a kind of nuanced argument to have. And it's not quite the same as entropy, because entropy just tells you about what you've lost and what options might have been available. It doesn't tell you about structures you can carry forward in time. And from operation to operation, which is of course important to this community because you want to build motifs and reuse those motifs again and again. So what assembly theory is, it describes the minimum number of physical steps needed to construct a given object. Assembly information is a function of the abundance of the object and the assembly number. So what I mean by that is if I say I was going to make C60, the way I might make C60 is I take a carbon atom, and then I might repeat that carbon atom into, say, a pentamer, so 5 times repeat. And then I'll take those pentamers, those pentagons, and tile them with a hexagon to make the overall structure. And I won't need to, it tells me how I can use the symmetry of those carbon atoms, of course, and I understand the point group of C60, and it's just a way of expressing how I would enumerate those in time to build the object. The higher the assembly number, the lower the number of objects that will be accessible using random events. So all I'm saying is that the C60 has a very low assembly number. It has a very high symmetry, easy to make. Vitamin B12 and Taxol, very high assembly numbers. You need information to make them. You can't randomly get there. Or you need some contingency in the environment or evolution. So there are some good applications of assembly theory. When you invent a new physical theory or a new math theory, there's lots of new math theories. Is it physical? Can you use it for anything? Well, you can use it to compress stuff. You can use it to explain addition chains. You can use it to explain how you can compress text or images. What I'm going to talk about here really is molecules, which are a kind of graph or network. So to explain this in terms of a molecular context, on the left-hand side, I've got the building blocks of an arbitrary 2-dimensional object, and then I've got the letters. But really, I want you to look at the formation of the molecule. Every time I add a bond, and that bond is kind of used to construct a motif that isn't already known, I count that step. And on the right-hand side here, I just really express the probability of that happening randomly. And please forgive me I can forgive myself for talking too fast, but please forgive me for talking too fast and saying, look, when I take a molecule and I add a bond, I'm really adding a bifurcation into a tree of decisions. And the more bonds I add that are different, the more complex the tree, and that's what I'm trying to say. So the take-home message from this slide is, once you've added 30 bonds, well, 15 bonds actually, the assembly index is shown down here, the likelihood that you're going to make more than one of those objects is about 1 in a mole. So what do I mean? If I find an object that has an assembly number of 15, the chance that that will form randomly on just a random walk on a tree would be 1 in a mole. So if I find a milligram of them, I know something dodgy is going on, or there's some predisposition, that the chemists call it. I call predisposition magic, because there's information in predisposition. It can't happen for nothing. So we investigate a chemical space, the amino acids, and you can see this is probably perhaps a little bit more useful. I can see that as I build up asparagine or tryptophan and then penicillin, and then at the top here I've got ATP, and you can see how I build up ATP in terms of its graphs to make the final structure. And something I should add at this moment, this has nothing to do with synthesis, which is kind of odd given I talked about synthesis and the information required to make a molecule. This is the worst case scenario, or the best case scenario if I'm thinking about graphs. Like if I'm thinking about drawing graphs, what is the easiest way to get there on a random walk on a tree of bond formations? And on the bottom right corner, I show here that how molecular weight tracks with MA. And then as the molecular weight kind of gets to about mid 250, it really explodes. And this molecular complexity measure is not dependent upon any weighting scheme or any information about the atoms or the kind of substructures that all the other complexity measures are. And that's why we invented it. We invented it because it's measurable. And this is the first complexity theory that ever has a proper objective measurement. And we showed this by doing infrared, NMR, and mass spec. And crudely put, the more parts the molecule has, the higher the assembly number. So if you take mass spec and you fragment the molecule, as it falls apart, you can have a correlation between the number of parts that you see and the assembly number. So we went and actually tested it on hundreds of molecules. We also did infrared and NMR and showed that molecular complexity is both intrinsic and quantifiable. It's universal, and we're applying it to drug discovery and looking for aliens. That was one of the reasons we invented it. If you go to Mars or Europa or Titan with a mass spectrometer, you won't know what molecules you're going to encounter. So you better be able to kind of work out if something is complex just based on the intrinsic fragmentation. And that we needed to do that before we were applying any machine learning, because we needed a ground truth. We need a physical theory. People applying machine learning without a ground truth drive me crazy, because it's just magic on magic. It's like teaching alchemists to do chemistry when they don't know atomic theory. And we need to have some kind of ground truth for that. But that's kind of a segue. But the nice thing is, once we've done this assembly theory, we can go into assembly spaces. And this is, if you're a bit bored and not knowing what I'm talking about, thank you for bearing with me, because hopefully this will start to converge in what might be of some interest to this community. So let me just quickly recap what I've said in parts 1 and 2. In part 1, I said, okay, reactions don't exist. Molecules are invented by reactions, and we have to invent those reactions in the environment. They don't just spontaneously emerge. The only way I know this has happened is through the process of evolution, and we don't know how life got started. But nevertheless, we can find complex molecules, and we can quantify their complexity using spectroscopy in an objective way. And here I'm just showing on this slide that we can actually quantify the complexity of some really horrendous looking molecules, like parataxel and mitotoxin, where you have this is just a humongous molecule. But actually it looks more complex than it actually is, because there's a lot of repeating motifs. What we've had to do in the lab is build some assembly calculators that have used Monte Carlo approaches to go through the space, because this is a very hard mathematical problem. But having said that, using assembly spaces, we can explore chemical spaces and overlap them. So say, if we find a pathway that makes aspirin or tyrosine, we can look at shared pathways. So as we share those pathways, we can make those molecules more efficiently if we're making them on their own. We can also apply that to ATP, as shown on the right-hand side here, and how we can take those building blocks and go through key step 1 and 2 and 3 and see how we invent ribose, adenine, guanine, cytosine, uracil, thiamine, and AMP, and then also get all the way up to ATP. So in that network of inventions, we are inventing information, if you like, or storing memories about the past, but able to produce those. And then in the final slide or two, I just want to kind of show how we can think about this pathway in terms of a bigger picture of going up a molecular tree of life, if you like. And what assembly theory is trying to say is that to if you see complex molecules, obviously the kind of, I suppose, the pinnacle of those is going to be a living system with some genetic processing going on. The system that invents those has to store the information somewhere. And that's a really interesting question for this community if you're interested in the origin of the genetic code and how polymers can suddenly build themselves. And there's a big question about how we might be able to invent new deterministically programmable systems, as some of the systems you're playing with are almost deterministically programmable. And then also, we're now looking at this to look at drug discovery. So there's a good example here of taking the tree of opiates and trying to guess what opiates are going to be invented by biology next. Imagine you're around in a billion years. What does the tree of, morphine and look like? And this is important for drug discovery because if we can run evolution forward, we might be able to come up with new molecules, pull them from the future to the present, if you like, if we do in silico kind of forward evolution. The other thing that's important, particularly in the US right now, is to look at molecules that are good at relieving pain, but they are not addictive. This is a really super hard problem, and we've been using assembly theory and the robots to explore new chemistry. And these two graphs here show that if we randomly generate molecules, or we use assembly theory to generate molecules, we can make molecules that are more drug-like than from the random distribution, which is hopefully obvious, but we were never sure if that was going to be proven. And then finally, before I stop, the most important thing that I think is interesting is that we can apply this to genomic information. To say, if I've got this kind of, you can now imagine like how many proteins can I make for a given sequence of DNA, okay, if I'm allowed multiple splices and resplicing. And this is a very well-known problem to this community. And I wonder if we can use kind of this kind of assembly theory to compress sequences in some way, if the unpacking of that by joining the parts together was effortless. Of course, it's not effortless, it requires some effort. But it is a kind of interesting thing to kind of imagine, and maybe we could use assembly theory to build very complex modules in biology, or understand how biology gets to modularity and robustness and uncover some of those regulations, if you like. Right, I'm almost at time. I promised to, although I was going off the beaten track, to be on time. So I'd like to conclude by saying that chemical reactions are invented, not obviously by magicians, they are invented by selection. So that's, I think, important. And I think the DNA nanotech community understands the systems you're playing with are invented by selection. So how do you manipulate selection to get yourself into a zone where you can do engineering? And that molecular assembly theory can be used to quantify how assembled a molecule is. This also applies to polymers and aggregates from them. And the assembly number of molecule can be experimentally measured. This is super important if we're thinking about being as objective as possible in exploring chemical space. And there are universal applications to physics, computer science, and biology. And I'd like to stop by thanking the organizers. I think you've done a brilliant job. I was making fun of them, sending me emails, but those emails were helpful because it made me get here at the right time and find out what was going on. So that was it's really hard running a virtual meeting. And I really do want to thank them genuinely for that, because bringing us together at this time has been really satisfying. And I've really enjoyed listening to all the talks. So, I really am very grateful to both of them for doing such a tremendous job. I want to thank my team. I think this is maybe pinnacle of the team in Glasgow, because with all the funding cuts and COVID and the fact that I'm this is all, I don't know what's happening. It's all a bit annoying. I've not mentioned Brexit today, so. But anyway, I'm very privileged to have this team working with me at the moment. I've got a very large amount of funding that's allowed us to do a lot of this stuff, and it's been a pleasure to explain some of this to you today. And I hope that I wasn't too annoying and a little bit provoking, and I'll be happy to talk in the channel for questions later. Thank you.
Thank you very much, Lee.